That's perfect. So welcome, everyone. We have a very exciting session on biomarkers and RCC. It's very nice to do this, especially after a very moving tribute to Michael, who's done such phenomenal work in the field. He had a comment yesterday about biomarkers in kidney cancer that sort of stuck with me. I think there was something like, you would look this up on PubMed, I think 144,000 papers on RCC biomarkers. And we don't necessarily have one that's stuck perfectly yet, but there's this tennis player back in the 1970s, Vitas Gerulaitis, who lost 16 times in a row to Jimmy Connors, and then finally beat him on the 17th time. So nobody beats Vitas Gerulaitis 17 times in a row. And that's how I feel about biomarkers, that each of these times we've made steady progress, we've made incremental improvements, and largely due to the people on this stage. So really excited for a very action-packed session. To start off, there's no one better to do this than Dr. Samra Turalic, who's going to speak to us on promising biomarkers, challenges, and opportunities for clinical implications.
Thanks very much, David, and thank you to the organizers. It's always such a pleasure to come to this meeting because of the many friends and colleagues in the audience, and I'd also like to extend my congratulations to Michael and thank him for all his work. And actually, I think it was a perfect introduction to this session, and let's see if we can get to that 17th win. So before we jump into the topic of biomarkers, I just thought it'd be worth reflecting on the fact that we often discuss biomarkers in a very reductionist way. They are, in fact, biology. They reflect biological processes. And if you're doing biomarker research, you are doing biology research. And biological processes, of course, can be very complex, and therefore it's not likely that a single biomarker is really going to reflect all that complexity that we see in the clinic. They're also dynamic, and so the dynamic change itself can represent a biomarker. You all know I'm a little bit obsessed with clonal evolution, and therefore biomarkers, as a result of cancer evolution, can be very spatially variable, and of course that gives us the sampling bias. And finally, biomarkers are context dependent. The same biomarker can mean different things in different tissues and in different patients. And so we can't expect biomarkers to deliver if we ignore this enormous biological complexity. Now, I was given this topic, and actually at ASCO GU, I was given a very similar topic, and that was a very prescriptive presentation, and that's not what I want to do today. But if you wanted to look at that, it is featured on Neuro Today. What I wanted to do today is focus more on some conceptual topics around biomarkers. So one will be sampling bias, and the other will be accounting for that complexity. So how can we begin to really capture it in a way that's going to facilitate personalized medicine? So one part of that will be learning from patients, and the other will be digital twins, and I hope this speaks to some of what Michael was alluding to earlier. So cancer is an evolutionary process, and it goes through all these stages of evolution as it progresses to metastasis and treatment resistance. And as a result, tumors are not homogeneous. They're subpopulations of tumor cells that are related to each other, but distinct in many different ways. And that also applies to the tumor microenvironment. And so as a result of that, our biomarkers, but also our drug targets, or the mechanisms of resistance to the drugs that we're giving, can be limited to a subpopulation within the tumor. And then that, of course, means that our biomarker or our drug is likely to fail because it's not addressing the tumor comprehensively, or we're really stratifying patients by something that is not representative of that patient's tumor as a whole. And there are good reasons and examples of how this leads to failure across many therapies, not just in kidney cancer. But what I would like to argue today is that measuring this complexity and capturing this landscape is where our problem lies. And we have several levels of undersampling, tumor microenvironment, and the host itself, things like immune fitness of patients. I mean, this is barely ever sampled, and yet we know it can profoundly influence how therapeutic response is. Furthermore, I would argue that we have a problem with sampling technologies of the tumor. So we've seen this huge pace in advances in molecular testing. So today you can order a clinical whole genome sequence, not in kidney cancer, because unfortunately we still don't have actionable alterations, but in many other cancer types, you can get a routine whole genome sequence, and that really reflects this amazing leap that we've had over the last few decades. At the same time, the way that we sample tumors has not changed at all for decades, and yet most of our patients will have tumor sampling at some point. What I really want to highlight here is that the tests that we do are performed on cancer samples, not the whole cancer. And I really want to emphasize how profound that undersampling can be, especially for very large tumor. So a single region biopsy contains about half a billion cells, while a representative sample, especially large kidney tumors, they'll contain hundreds of billions of cells, sometimes over 1 trillion cells. Now, if this was a blood cancer, it would be fine, because every time you take a blood draw, you are getting a well-mixed population, so you get a representative sample, you find everything that's there. But the problem with solid cancers is this mixing of different populations of cells does not occur. It's very spatially separate, and therefore, every time you put your needle somewhere, you are going to get this spatially separate information. So just a quick example here, again, to bring this home. We don't have predictive biomarkers that we use, but let's just take CA9 staining. So you might do this on around 300,000 cells as a pathologist. And let's say you're in the US and you're David, you can order a Foundation Medicine test or something else. You might do this on around 3 million cells. But if you look at what's actually sampled from the entirety of the tumor, it's really tiny. So we have an issue with sampling. Over the last few years, we've decided to broaden this sampling frame, essentially by taking over leftover surgical tissue. This is after sampling by the pathologist. And what I want to emphasize is that this is tissue that's normally incinerated. So once the pathology report has been signed off, this tissue and information that it contains is destroyed forever. And what's important is that we take this tissue over, we homogenize it, so we get over that spatial bias. And what we've shown is that then applying a molecular profile to this type of sample gives you a much more reproducible and much more representative readout. Over the last couple of years, we've improved this method by introducing another step, which is flow sorting of tumor cells away from stromal cells. So we are now working towards a representative readout of the tumor microenvironment and the phenotypes that are there. At the moment, we're taking this forward as a feasibility study in a clinical trial, and kidney cancer is one of the subtypes we will be looking at. So I look forward to reporting on this in due course. Now, coming back to that complexity, we have a special challenge in RCC compared to most other cancer types. We don't have oncogenic targets. All our targets are in the tumor microenvironment, the vasculature, the immune infiltration, and of course, the interactions between all these factors are very complex. They're subject to some host factors that we don't know about. And most importantly, they're subject to dynamic change. So they're not going to obey the kind of elegant laws that you might have with those targeted therapies where your molecular target is on the tumor cell and your drug is perfectly fitting it. So really the reality is, and Michael pointed to this, we're using combinations largely empirically. So how can we do better within this? We need to consider the mechanism of action. So, of course, the vascular responses to VEGF modulation. The cancer immunity cycle presents a greater challenge because of many aspects of this and the different mechanisms of action of different immuno-oncology agents. And also, we don't really know which aspects of these biological processes matter most in the biomarker context. So this is where the biology has to converge onto something that's measurable and that's stable enough. So I would argue that the best way to do this is to measure the activity of the drug in action. And I don't mean this in a preclinical model, I mean in the patient, because that is the ultimate model that we need to understand the ground truth from. And we've done this successfully in others, in these labor of love studies, and it's a labor of love for the patient, it's a labor of love for the physician and the scientist. The patients undergo on-treatment biopsies at progression biopsies. And for those patients where the treatment fails, some of them have also taken part in autopsy studies, and we're very grateful to them and their families for allowing us to extract this knowledge that I think has probably been the most valuable thing that we've done, certainly in my translational research program. This is a more recent study, although, again, we started recruitment into this study about 10 years ago, looking at the impact of VEGF-TKI treatment on the immune tumor microenvironment. So this really can underpin what we need to understand about the interaction between these agents that we're combining in the clinic. Of course, the holy grail is using biomarkers to stratify patients, and some wonderful examples from colleagues in France and US who are actively doing this. Another opportunity to understand if we don't have the clinical trials, are the exceptional responders. And congratulations to David on this beautiful work earlier this year on tertiary lymphoid structures. Again, this is something that we are seeing, and Zed Tipu had alluded to this data in his presentation, and I think we're going to hear about this in this session as well. So the last challenge I want to talk about is how we rationalize these combinations and how we contextualize the host. That's I really believe this is the last barrier. So we can't carry on as we are. There are thousands of combinations in trial across oncology, not specifically in kidney cancer. And as we know, there are often not enough patients who meet eligibility criteria for these trials to even recruit into them. The host factors, things such as microbiome, the immune fitness of the host, the interactions with the germline genetics, these are not measured and they're not known, and yet they can profoundly influence how the therapy is working and lead to different outcomes in different patients. So I think the opportunity here is really about merging data, mechanistic models, and predictive modeling. And this is going to accelerate not just our biomarker research, but our drug development as well. So having a digital twin is something that represents very detailed data on a patient that also provides dynamic information on treatment. This allows us to realize the potential for precision medicine. When we do this across many patients and we amalgamate the data at scale, we can create a generic patient model. These data can replace control arms and synthetic in real-life trials, and they can account for unseen scenarios. But this needs to be underpinned by carefully curated data, harmonized data, sampling, workflows, etc. And this is where I'll just skip to my final slide because I'm running out of time. But I just wanted to tell you about this platform that we've launched recently in the UK to achieve precisely this. Harmonizing sampling and assays, integrating multi-omic data at scale, and also testing these nascent new technologies where there is biomarker information, but we don't know how it's going to fit in. What's really important here is this iterative process. Our patients go through multiple lines of treatment. The biomarker status and the meaning of the biomarker is going to change every time with that change of context. So these models, the predictive models, need to be iterated through this continuous feedback from patients. Some of our Manifest Consortium and partners are in the room. This really takes a large effort. We're working on this across the UK, but we would love to work with colleagues elsewhere. This is what the platform is, and actually, if you want to find out more about it, this is fresh off the press in Cancer Discovery today. So my take-home messages are, insist on translational points in your trials. We owe this to our patients, and we owe it to ourselves and all the hard work that's gone into the work today. We need to harmonize and collaborate. This needs to be happening internationally. And if you are thinking about new biomarkers, you need to consider implementation in advance, what the path to translation is going to be. We have to campaign, and I think this is where KCA could do good work, and all patient advocates, to improve data and sample access from industry-sponsored trials. And it really takes a village to do this kind of work. So on that note, it's a great privilege for me to work with many talented scientists at the CRIC and clinicians at the Royal Marsden, but most of all, I want to acknowledge patients and their families. Thank you. So it's my pleasure to introduce and welcome my friend and colleague, Tom Mitchell from University of Cambridge. Welcome, Tom.
Perfect. Thank you very much, Samra, and thank you very much to the Scientific Planning Committee for tasking me with this talk on next-generation sequencing, unlocking new insights in kidney cancer. For those of you that don't know me, I'm Tom Mitchell. I'm a urologist in Cambridge, but a group leader at the Early Cancer Institute. And I'm going to be talking about primarily how sequencing can better inform the growth of localized kidney cancers. And I'll run through the existing surveillance strategies, what we use for small renal masses, and how these can be improved, A, via germline genetics, but also by tumor subtyping. Before then, I'm talking about how somatic mutations may improve these models. And I thought it'd be useful to frame some of the issues in a recent case study for a patient we saw in Cambridge who presented in his 70s with comorbidities, with a small renal mass, and it'll be no surprise that after shared decision-making, we went on with surveillance. He had scheduled scans every 6 months' time, and 2 years later on, there was no significant change in this mass. His next scan, 8 months later, we see it's grown by approximately 5 millimeters in that time. He went on to continue to have surveillance, but unfortunately 1 year later, the mass now measures 4 centimeters in size. There's lymph node involvement with adrenal and pulmonary mets. And clearly this is an uncomfortable scenario for lots of us in that it appears that we have missed the curative window. And the question is, how unusual is a case like this? And if you look to the large data cohorts available, either from the DISARM registry on the right or the Finelli Jewett data on the left, we see that it's not that unusual in a static snapshot, in that almost 1 in 10 patients may have growth rates of more than 1 centimeter per year. But unfortunately, we don't have data on whether a low growth rate to start with will equate to continual low growth rates. If we look to our surveillance regimes, it appears that that is the case, in that most recommend quite intensive surveillance to start with, and then with timelines lengthening as time goes on. But you can ask the question, if we were to subtype these tumours better, is there anything molecularly that could improve things? Would that help? And of course, we can look at inherited RCCs, and we get a bit of data from this. This is from Ball et al. In inherited tumour syndromes, and he showed that if you have an inherited BAP1 mutation, then your BAP1 deficient clear cell tumour will grow faster than a VHL deficient clear cell tumour, and they will grow faster than the cohort of other tumours, either MET-activated papillary RCCs or folliculin deficient oncocytic neoplasms. If we look in the sporadic setting, there's less data on this, but in the Finelli dataset, we also see similarly that those tumours that are clear cell in origin seem to grow faster than papillary and chromophobe tumours. Based on this, we can ask actually whether, if we knew the specific mutations within a tumour, whether that could infer how quickly or not a tumour is growing. And so if we use the example of clear cell RCC, we know the evolution quite well from the TRACERx studies and others. We know that the first hit in these tumours is 3P loss. The second hit tends to be VHL mutation. And after that point, you have your clinical tumour formation. And then you can draw from any number of subsequent events, PBRM1, BAP1, SETD2, or chromosome losses. And they will confer a selective advantage to that cell, which will then outgrow the tumour. And so we asked ourselves what the impact of these mutations might be on tumour growth. So we've been assembling a cohort of patients who've had longitudinal imaging prior to surgery. This is between UCL with Maxine Tran and the Cambridge PI of artist, James Jones. And when they go on to have their surgery, we have multi-regional sequencing. We can look at the radiomic features, and we look at clinical outcome, and we can see if we could have predicted what happened to these patients. And if we look at this data, we see that the tumours with the minimum driver events, so 3P loss and VHL, those are shown in green here. They grow the slowest, so by 2 to 3 millimeters per year, the expected growth rates, whereas every subsequent hit you have in these next events, we see faster and faster growth rates. If we were to split this data and do it by size, we see a number of these tumours which are small renal masses and therefore perhaps indolent. Actually, if we recapitulate the growth rates, they are in fact growing very quickly, and they have these additional mutations. We can also use this data to quantify the effect of each individual driver event on growth rate. And we see here that the perhaps more indolent mutations, PBRM1, we see an additional growth of about 2 millimeters per year. But if you look at the most aggressive subtypes, say if you've got a BAP1 mutation, that's going to increase your growth by almost 10 millimeters a year. And this matches what the Ball et al. Paper showed in the inherited setting. And so there's a strong effect size in terms of the mutations in tumors, but of course we see a large variability. And this is something that we've touched upon a lot this morning, in that there's the microenvironment and there's patient-specific risk. And if you look at the causes of kidney cancer, particularly clear cell kidney cancer, there's a contribution from germline genetics of perhaps up to 40%, large contribution from obesity, and a significant contribution from smoking. And in general, none of these processes are mutagenic. Now, there's a small contribution from smoking that we saw from the mutagraphs studies. But therefore, all of these risk factors must increase the rate of growth of tumors. They must increase the fitness of that cell that's gone on to have a VHL mutation, PBRM1 mutation. And so you can envisage a schematic such as this, where if you've got the baseline drivers, you'll have a baseline growth rate, but say if you're obesity, you're going to have a faster growth rate. If you go on then to have a BAP1 mutation, your tumor is obviously going to grow a lot faster, but if you're a smoker, for instance, then that rate and that chance of metastasis will be so much higher. I'm going to leave it at that. A couple of take-home messages. Surveillance of small renal masses is still safe. I'm not disputing that, but subtyping by biopsy can help, and that's only just by working out. That's not only just by working out if a tumor is benign or malignant. We believe that growth strongly depends on the driver mutations that you see, but there's a big caveat. Predicting future growth will be challenging, so there's patient variability, but also there's the chance, perhaps, in the same as the case study we showed, that there may have been acquisition of an additional driver mutation which was unforeseen. I'll leave it there. I'd just like to thank a few people in my lab, Peter Campbell and Grant Stewart, Adam Brooks, and of course our funders, but most of all our patient donors. Thank you very much.
Thank you, that was wonderful. So next we have Aria Kiwi coming from Memorial Sloan Kettering, who's going to talk to us about the tumor microenvironment, key insights, and implications.
Thank you very much, and it's my first time in Amsterdam. I'm just blown away by the beauty of the city, so wonderful host city. I'm tasked today by talking about the microenvironment, which really dovetails nicely in with the other two speakers, and hopefully will spur some discussion later on. I think we know now from several large-scale studies that RCC is made up of things that Samra was alluding to, not just tumor cells, but in fact, in many cases, bulk of what's seen when you separate out the components is made up of non-cancer cells. And some of the early work kind of really set the stage for what's been validated in multiple single cell and flow cytometry studies subsequent. But essentially, these are tumors that are comprised, at least of the clear cell, of a lot of T cell populations, followed by mostly myeloid populations, which are comprised of tumor-associated macrophages, neutrophils, and other non-T cell populations. And then, of course, there are other rare populations that are seen across. But across and within patients, there is a tremendous amount of diversity, and that can be useful in understanding underlying biological principles, but also poses major issues in terms of biomarker development, as was also discussed earlier. And so some of the work that we've published in my lab, in my group, has kind of focused on this concept in the context of immunotherapies, which obviously I think drive the majority of our success in the metastatic setting. And if you look both within and across patients, you can start to get the sense that T cells themselves may be associated with responses. We know that if you look just historically across solid tumors, that having a lot of T cells in the tumor does not necessarily mean the tumors are going to be less aggressive tumors. In fact, there's some data suggesting, at least in kidney cancer, that high T cell infiltration, just looking at it as a single marker, may be associated with more aggressive tumors. And that's really because we haven't phenotyped the T cells properly when you're looking at those types of contexts. But in the context of therapies and using single cell approaches, you can sort of get a sense of what types of the T cell populations are there and the myeloid populations. And when we do that type of work, we can start to see, are there specific populations that are associated with responses and resistance? And at least we're starting to get the sense that long-term immunity, long-term responses, which is essentially what we want to go with, what the home runs would be, are really indicative of sort of T cell memory responses. And as we start to develop those types of things, we can start imagining how that might play out into the biomarker space. We also know, though, that these populations will evolve in the context of tumor development as well. And work from David Braun and from Ellie Van Allen and Tony Chouari and others have demonstrated that the T cell populations and the myeloid populations will co-evolve as tumors become more invasive and more aggressive. And these populations may also be relevant to understand, as was what Tom was describing, the nature, the indolent and yet sometimes aggressive nature of the small renal masses, how tumors evolve themselves and start evading immune responses is also intimately related. So there's a clear need to continue to understand, both on a mutational and genomic level as well as a microenvironmental level, what is happening both in the early stages as well as in the advanced stages. So I'm just going to briefly take us through where we currently stand in terms of understanding this concept from a pure biomarker standpoint, and then I'll talk about some work from our lab more recently that kind of suggests potentially a way forward, at least with respect to other populations that we're not currently targeting. So we know from a lot of the work that was done out of the seminal series of papers that came from the unsuccessful trial in terms of therapies, but super successful trial in terms of advancing our understanding of the tumor microenvironment in clear cell renal cell carcinoma, was the work from David McDermott and others in Genentech. And this sort of kind of emerged the concept that we have sort of dominant subgroups of tumor microenvironmental tumors. Using RNA sequencing in this study, they identified angiogenic signatures, what they call T effector signatures, and a myeloid response signature. And again, these are sort of vague terms that are used to capture, I would say, cell states or microenvironmental states. And you can see that there are tumors that are very angiogenic at baseline. These are often associated with PBRM1 mutations. They're more indolent tumors. You can have tumors that are very myeloid-driven. These are generally very immunotherapy-resistant tumors, but also resistant to VEGF therapies. And then you have these T-cell infiltrated tumors that lack sometimes the myeloid responses, and these tumors tend to do quite well in terms of immunotherapies. And then this has sort of been merged together now with some of the mutational work, especially work from SAMRO, integrating some of the TRACER-RX kind of concepts, even though they're not necessarily the exact same subgroups, but sort of taking the key genomic groupings and layering on these microenvironmental features and sort of coming up with different clusters. These have now been applied into different clinical trial data sets. I will say this is an emerging way of thinking about the tumors. It doesn't necessarily correlate very well with response, but we do understand that there are these kind of states that exist with both mutations and the subsequent microenvironmental understandings. All of this being said, that we are sampling 1 or 2 regions of tumors to do these types of work. So Samra's points earlier certainly come into play here in terms of understanding this broadly. And that, that is something that we are also thinking quite a bit about. And these signatures have also been used, though. These T-cell signatures have been applied to certain individual trials. This is the work from Bob Motzer and others from the Javelin Renal 101 trial, looking at some of the T-cell signatures and some NK signatures. Again, these are nicely associated with response on individual trials. But if you if you look at some of these signatures that have come from different trials, they don't necessarily validate across trials, which is why I think we certainly have a lot more opportunities to refine what really matters in this context. And that's where I think things like single cell, but actually more likely spatial technologies will help us really understand what are the key components of these, and can they be reduced to immunohistochemical markers as opposed to the need to do next generation sequencing, which is probably overkill for a lot of these kinds of questions. But essentially, this is sort of where we're at. And this is the emerging data from the CLEAR trial, which was the lenvatinib pembrolizumab trial. This was a trial where most of the patients had an initial response, which is great for patients, challenging for biomarkers. If everybody responds, how are you going to ferret out the ones that don't? But what you saw in this trial, and not all of this was, they actually couldn't find any biomarkers. Angiogenesis did of course, predict response to the control arm, the sunitinib arm, but the other signatures weren't necessarily as reliably predictive in this work. And so that suggests that, not all of these may not a single biomarker may be useful across the different trials. But what it also brings up is the notion that what you're pairing with may also be relevant. So the TKI partner may also be driving a lot of the response that we see as we try to compare PD-1 TKI trials across from each other. And David Braun has recently shown that we can look at extreme responses and try to understand this. And I think the concept of TLS is may be something that we can certainly use and may be a useful biomarker, although I will say that true TLSs are relatively rare in kidney cancers, but smaller pockets of T follicular helper cells and B cell infiltrate may be existing a lot more than we think when we're starting to look at this now across spatial technologies. So we do have opportunities. In this particular study, they found that at least with respect to VEGF TKI-IO combinations, they saw that TLSs were quite associated, and these more memory responses I mentioned earlier that we saw in our single cell work were more associated with these extreme responses to PD-1 CTLA-4, and that may explain why a true memory response is associated with potentially curative states that we see that are really restricted mostly to the CTLA-4 PD-1 setting. And I think genomics is telling us what we are seeing clinically in this context. But I do want to also take a minute at the last minute I have to talk about how the other populations may be relevant. And I think something that came out of work that we did many years ago now on first-line TKI therapy, so these are the earlier drugs, but what we did find was when we compared microenvironmental determinants of response to pazopanib versus sunitinib, we actually saw quite different, if you look at the curves on the right, quite different responses with respect to pazopanib and sunitinib. And keep in mind, these TKIs are not clean. They're not just targeting VEGF. They're targeting many other populations, including kinases that are present on macrophage populations. For example, sunitinib targets CSF1R. And so there's opportunities to kind of understand what the partners are in terms of what they're targeting here. And this is sort of the work that we're doing now. This is in review, led by Linda Vuong from my lab. And we're kind of combining using mouse models, both syngeneic models and GEM models, to understand kind of what's happening with the microenvironment when you're treating with PD-1 alone or with the TKI alone or with the combinations, and understanding what is actually being shaped by something like lenvatinib, for example, and how that might be associated with human response as well. So we're leveraging the notion that we're operating a lot later on in the spectrum of disease now. Thanks to things like Carmina and Surtime, we have moved away, not entirely from upfront cytoreductive, but certainly we're doing a lot more consolidative surgery later on. And that allows you to look at these populations in the context of treatment, in long-term treatment, right? So the unique thing we do in kidney cancer is often these patients will get 6 to 9 months to 12 months of treatment. And that allows you to look at the evolution of populations, and we can go back and forth between the mouse and human and understand what's happening from the TKI perspective. And then we can start saying, can we refine signatures even better now, taking these dominant myeloid populations that are being affected by TKIs and determine who's going to respond and who's not? And does that apply across trials? Does that have to be restricted to the individual TKI you're using? And I think we will get to a point in the next several years where we will be able to start doing this, and maybe we can use that up front to determine what sort of TKI you might want to partner with a PD-1 in that particular patient, or what should be next after they progress on those therapies. So there's a lot of opportunities there. I would say that the TMA is clearly associated, the take-home is that TMA is clearly associated with response. We don't have kind of validated biomarkers across trials yet that we can certainly say this is what you should get. But I do think that the myeloid populations will be the next frontier in terms of what we do target, in addition to T-cell populations, to try to get to those home runs that we're all searching for. So thank you very much for the opportunity.
I think we've been hinting at this next talk quite a lot on TLS as an emerging biomarker in metastatic RCC. And it's my pleasure to welcome an early career researcher, Anna Calves. Over to you.
Thank you very much for the kind introduction, and thank you for the invitation. So I'm very happy to be here with you today and to share our data on tertiary lymphoid structures and emerging biomarkers in metastatic RCC. So immune cells in the tumor microenvironment can be organized into tertiary lymphoid structures, or TLS. These structures are organized immune aggregates, which are mainly composed of B and T cells, and they are the sites of a local anti-tumor immunity generation. They are important because their presence inside the tumor microenvironment has been associated with a favorable prognosis and better response rate to immunotherapy, and especially to immune checkpoint inhibitors. And you can see here in this study that was published in Nature Cancer in 2021 that includes more than 500 tumors and 18 types of cancer, among them RCC, that patients having TLS, and especially mature TLS, better respond to ICI and have a prolonged survival. However, not all patients with TLS-positive tumors do respond to immune checkpoint inhibitors. And you can see here in the same study again, that was nicely published this year, that a part of the progressive disease patients do have high TLS score, meaning that these patients probably have TLS inside the tumor but do not respond to immune checkpoint inhibitors. So this led us to the hypothesis that there might be mechanisms that are involved in the inhibition of the anti-tumor activity, which is implemented inside this TLS. And to address this question, we are using the Bionic cohort that includes 199 patients with metastatic ccRCC. From these patients, bulk urine-seq was performed on the primary tumors of 92 patients that were then treated with ICI or anti-angiogenic treatments. And we then stained the tumors to identify the ones that were TLS positive, and we focused on the 58 TLS positive tumors that were then treated with ICI. So to understand the non-response to ICI in these TLS-positive tumors, we used this bulk RNA-seq data, and we performed a differential expression analysis to compare the TLS-positive tumors from responders and non-responders. And as expected, we found many signatures that were upregulated in responding patients, such as B cell activation and differentiation, Th2 cell activation and differentiation, macrophages activation, and this supports the implementation of an anti-tumoral immune response in the responding patients, which makes sense. However, what was surprising to us is that the top signature that was upregulated in the tumors from non-responding patients was this GABA transport signature. And when looking at the other signatures that were differentially expressed, we found that this GABA synthesis signature was upregulated in non-responding patients as well. So the high expression of these GABA signatures in the TLS-positive tumors is associated to non-response to ICI. But what is GABA? GABA is a gamma-aminobutyric acid. It is a principal inhibitory neurotransmitter in the adult brain, and it is normally synthesized in the central nervous system. It is essential in many functions, such as normal brain function, neuronal activity, and information processing. And recently, it's been described that this pathway could be reactivated in a tumor context. So we kept working on this association between GABA and non-response to ICI in these TLS-positive tumors. And so to do so, we used a reduced GABA signature, and we divided our patients into groups based on the expression of this signature in the tumor on the secondary side. And you can see that 85% of the tumors that have a high expression of this GABA signature are from non-responding patients. And 74% of the tumors that have a low expression of this signature are from responding patients. We also checked the survival, and we found that patients having GABA high tumors, here in yellow, have a shorter progression-free survival. So the expression of this GABA signature is associated to a higher proportion of non-responding patients and a shorter survival. And we wanted to confirm this in other CCRCC cohorts. And indeed, we found the same association between GABA and non-response to ICI in the TLS-positive tumors from the nivolumab cohort that includes 82 patients that are then treated with nivolumab, but also in the Emotion 150 cohort that includes 31 patients that are then treated with atezolizumab, and in the Emotion 151 cohort that includes 174 patients that are then treated with both atezolizumab and bevacizumab. So the expression of the GABA signature is associated to non-response in two different ICIs in the TLS tumors from several CCRCC cohorts, so Bionic, Nevorin, Emotion 150, and 151. However, this effect was not found in the TLS negative tumors or in the tumors that were treated with tyrosine kinase inhibitors regardless of TLS presence. So this association between GABA and non-response to treatment seems to be specific to TLS-positive tumors that are treated then with ICI. And if so, we wanted to understand which cells could produce the GABA inside the tumor microenvironment. And so we used CCRCC single nucleate data that were published last year in Nature Genetics, and we checked the expression of the GABA synthesis signature in every cell type. And we found that the GABA synthesis signature was expressed in the malignant cells. So the tumor cells are the ones producing GABA inside the tumor. But are all tumor cells producing this GABA? To answer to this question, we used the same data, but we focused on the tumor cells. And we performed a clustering that allowed us to get 5 clusters. We checked in each cluster the expression of the GABA synthesis signature. And as you can see here, the light green cluster is the one with the highest expression of this GABA synthesis signature expression. And so this cluster is a cluster that has a transcriptomic profile that is close to the proximal tubule cells in healthy kidney. So we called it the proximal tubule-like or PT-like tumor cells. And these PT-like tumor cells are the ones producing GABA inside the tumor. And because these PT-like tumor cells do produce GABA, and as GABA is associated to non-response only in TLS-positive tumors, we hypothesized that GABA might impact the TLS themselves or the cells that are inside the TLS. So we used spatial transcriptomics to compare the TLS that were close or far from these tumor cells that produce GABA, the PT-like tumor cells. We did this in 4 patients that had both TLS that were close and far, and we found many variables that were depleted in the TLS that were close to the GABA-producing tumor cells. If we focus on the variables that were depleted in the TLS that were close to these tumor cells in at least 2 tumors, you can see that we have a downregulation of T-cell cytotoxicity, antigen presentation genes, and IgG genes, among others, in this TLS. So there is a dysfunctional immune activation that is observed in the TLS that are close to these PT-like tumor cells that do produce GABA. Then we wanted to know whether we would find the same characteristics in the TLS of non-responders compared to the TLS of responders. So we use spatial transcriptomics to compare the TLS from 8 non-responders to the TLS of 8 responders. And indeed, we found a downregulation of the T-cell cytotoxicity inside the TLS of non-responding patients compared to the responding patients. We also found a downregulation of the antigen presentation signature and antigen presentation genes in this same TLS of non-responding patients. And finally, we found a downregulation of IgG genes also in this TLS of non-responding patients. So the TLS of non-responding patients also show a dysfunctional immune activation. And this is maybe due to the GABA-producing tumor cells, the PT-like tumor cells that could be close to them. So to conclude, the high expression of the GABA signatures is associated to non-response to immune checkpoint inhibitors and to a poor prognosis in these TLS-positive CCRCC tumors. GABA in these tumors is mainly produced by the proximal tubule-like tumor cells. And the TLS that are close to these tumor cells, as well as the TLS of non-responding patients, show a dysfunctional immune activation. As a main conclusion and take-home message, GABA is a modulator of ICI resistance in these TLS-positive CCRCC tumors, and it represents a potential biomarker and therapeutic target to enhance ICI response. And I would like to thank Catherine and Hervé Friedman, as well as Stéphane Houda, who are supervising this work, Isaias Hernandez and Cheng Mingsen, who are working with me on this project, and all of our collaborators that made this work possible. Thank you.
And to finish off the session, we have Annika Fandler, who's going to speak to us about organoids and other innovative model systems for studying RCC.
Thank you. Yeah, so I will talk about organoids now, and you probably all have heard about organoids by now, but I want to go back and talk a bit what are organoids in generally, and including what we mostly mean when we talk about organoid, patient-derived organoids. And then how can we use them in basic and translational research in kidney cancer, but also specifically what are the current limitations and what are potential strategies to overcome these in the future. So if we talk about an organoid, we generally mean it's a 3D culture that has been generated by a stem cell. That can be a pluripotent stem cell, an induced pluripotent or embryonic stem cell, but most frequently when we talk about them, we also mean organoids from adult stem cells. And that has been done for a range of different tissues by now. You can see that here. And when we talk about tumor organoids, then the stem cell is not as clear. But generally, they are regenerated from a specific cell over time, and that also gives this long-term culturability that we see. So if we look at this slide, how do organoids actually compare to other model systems? If we look from the left to the right, the systems get more complex. On the left side, we have very easy systems, easy to establish in the lab, easy to do experiments on, classical 2D cultures, but they are not really representative of the physiological complexity, and they also don't really recapitulate human physiology very well. So we go to more complex systems, but then the ease of maintenance really goes down, and it takes a long time to do these experiments. It's sometimes really hard to genetically manipulate these, or takes just a really long time. So human organoids are kind of sitting in between this, where they are still very easy to establish. They can be easily cultured in the lab, but they have a far more better physiology recapitulating tumors. And if we look at this, of course, there's a range of applications where we can use organoids. So first of all, if we look on the left-hand side, this is what most studies are doing today. It's about predicting therapy success in these organoids and then characterizing the molecular phenotypes and genotypes in these organoids to also use it for biomarker development and to understand the mechanism of why patients respond to a therapy. But we can also use them for more basic research. We can genetically manipulate them, and we also can grow them over a long period of time to see how tumors will evolve eventually and kind of understand evolution on a more mechanistic basis. And of course, we can also start incorporating cells of the TME, and then we'll talk about this later in the organoids to make them more complex than what they are now. So this is an example of a postdoc I did at the Max Delbrück Centrum in Berlin a few years ago. There we identified a cell population that was quite undifferentiated and was able to propagate xenografts over time. And when we seeded these cultures, either as suspension cultures or as this classical organoid cultures, we could see the suspension cultures remained very undifferentiated, but the organoids differentiated, and they maintain typical markers of kidney cancer in these organoids over time. And we used this to test therapeutic strategies targeting these undifferentiated cells. You can see this on the left-hand side, and we could see patient-specific responses in each organoid, and we could reproduce these responses in xenografts from the same patients. And of course, this has not only been done in RCC, but it has been done by now in a range of kidney tumors. This is an example from the work of Jan Trost, where they have done it in childhood tumors. But what really here is a limitation is that even for RCC, clear cell RCC, the numbers are still really small. So for other subtypes, we still need much more experience to learn how we can grow them in the same way, what exact features of the culture conditions we need to really represent all the subtypes. And then in kidney cancer, we can not only use patient-derived organoids directly from a tumor, but iPS-derived organoids have also been developed by now several years ago, 10 years ago. And these are really helpful if we want to understand germline diseases or understand function, for example, of VHL during early development of the kidney, and can use this as a good model. We can not only use tumor cells and then derive a patient-derived organoid, but we can also use the normal tissue and then try to understand how this tissue evolves into a tumor. And I brought an example here from gastric cancer that has been published 2 years ago, where they used gastric organoids, normal gastric organoids, and then they knocked out TP53 and APC, and then they watched what will happen to these organoids. And what they could see over time, that copy number alterations began to enrich in these tumors. And this was cultured over 2 years, so you can see that this is a long-term experiment to understand how these tumors evolve. And if you look at the specific copy number alterations, you can see that these repeatedly enrich in the organoids, and they also represent copy number alterations that you can see in the clinic in gastric cancer. So, but what are the major limitations? These kidney organoids really are just epithelial cells, tumor cells that you can propagate. They lack any form of an immune system or vasculature. And I think especially in kidney cancer, this is a huge limitation because we can't really model the therapies we are doing at the moment. And secondly, there's still a lot of room for improvement when it comes to the efficiency of PDO formation and really understanding what clones are selected in culture. So if we look at this, if you look at the left-hand side, I brought an example of bladder cancer. And there you have specific mutations that you can actually kind of target by changing the growth factors. So for example, in tumors with P53 alteration, you can use Nutlin to kind of only enrich for specific populations. And in tumors with FGFR3 amplifications, you can withdraw FGF and these tumors will continue to grow. This is a bit more challenging in kidney cancer because we don't have this clearly defined mutations that we could kind of use for this purpose. But what we saw is that if we grow cells in hypoxia, then we can really enrich for tumor cell populations and maintain them much better than normal populations that would otherwise overgrow these tumors. And if we look at the lack of the tumor microenvironment, there are a lot of different strategies now how we can incorporate specifically immune cells into organoids. On the left-hand side, if you come from a single-cell suspension of the tumor, you can use co-culture method, where after separating them, you take the tumor cells, the immune cells, and you co-culture them to see if how you can activate the immune cell to target the tumor cells again. This can also be done in microfluidic devices, in microorganospheres, but you can also use a fragment of the tumor and not dissociate it fully, but take these fragments and then culture them together, the tumor cells with the still intact microenvironment, to use this in short-term culture, which are specifically well-placed for therapy studies. This is an example. Actually, this has been published by Daniela Thoman a few years ago, where you can take fragments of the tumor, treat them, and then with flow cytometry investigate the activation of immune cells after treatment, for example, with nivolumab. This is also an example from us where we could show that indeed in a patient-specific manner. If you treat these fragments for 2 days with nivolumab, you can see activation of immune cells. We measured this with surface markers that indicate activation. And then in some tumors, you see that these T cells that already are placed in the tumor can be reactivated. And it has been shown that this is predictive also of a clinical outcome. With the vasculature is, of course, more complex. And this is an example also published a few years ago in kidney cancer, where the team used microfluidic chips, where they seeded the endothelial cells. They are marked in green with GFP, and then they placed organoids on top. And you can see on the right-hand side that these endothelial cells began to grow into the organoid and form a vasculature in these organoids. So these could potentially, methods could potentially be interesting when we're looking at TKIs and how they function in these organoids. Yeah, with that, I want to conclude. And the take-home message are that PDO cultures are patient-specific tumor models in vitro, and they have a range of applications that we can use. But we need to understand the limitations. And specifically, we need to start incorporating and understanding the TME within these PDOs to improve their application in kidney cancer. Thank you.
Thank you so much to all the faculty for brilliant talks. And we had David Braun's paper referenced 3 times, so it might as well have given a talk, David, I think. Are there any questions in the room? If not, I would like to start. Yes, please go ahead.
So Helene Knevsrud from Oslo. I have a question about this tumor heterogeneity, both for the sequencing and the patient-derived organoids. So what would you say is the best practice today to account for this heterogeneity?
Why don't you go? Yeah.
So for the organoids, I actually think it's challenging. So there is heterogeneity still within these cultures, and we can see that, but we are actually not really good in maintaining this heterogeneity and understanding it fully. We will always use some of the clones, and I think we're only beginning to understand what we need to add to the medium to maintain each clone, and what we're actually using and what we are keeping. So.
Yeah, and as far as patient tumors are concerned, the question about how many biopsies you should take is an unanswerable question, because there is a broad range of degrees of heterogeneity between patients. We don't have a priori knowledge of that. So I think approaches such as I shared with you is one way, but ultimately I think where the field needs to be going is blood-based measurements. Of course, we are already seeing that for tumor cell intrinsic features, such as genetic features. It's not been as successful in kidney cancer as in other cancer types, but probably the sensitivity will improve. But I think we also need to be measuring tumor microenvironment, metabolome, everything really from blood-based in the future. I think that's the only way to get the representation. Martin, go ahead.
Thank you all so much for your presentations. I have a question for Dr. Calvis. So congratulations on very provocative data. I have 2 questions for you. So for one, you must be spending a lot of time thinking about the mechanism behind GABA and how the pathway leads to immune modulation. Maybe you can share. And then secondly, have you looked at non-TLS containing kidney cancers and the effect of GABA signaling there on treatment responses?
Yeah, so first on the mechanisms. So it's described that GABA have immunomodulatory effects, and so we wanted to know on which cells it could have an effect in these tumors. We looked for the cells that were expressing the receptors for GABA in the tumor microenvironment of these patients. There are many cells expressing it, including tumor cells, but also myeloid cells and stromal cells. B cells also seem to express it. And so because B cells are mainly inside TLS and we are seeing an effect on TLS, we also focused on this. And we found that B cells that are cultured with GABA do proliferate less, and then when they differentiate into plasma cells, they produce less IgG and IgM. So there might be an effect this way. They also have a change in the metabolism, including the TCA cycle metabolism. So that's maybe one way. It may be going through different ways also. And the second question, I don't remember.
Without the TLSs.
Without the TLSs, yes. Thank you. So we've looked in CCRCC in the tumors that do not have TLS, at least in the pieces of tumors that we do have. And there was no differences in the expression of the GABA signatures between the responding and non-responding patients in Bionic, but also in 2 other cohorts. So yeah, it seems to be TLS specific, or at least in a tumor that do have immune activation. And so TLS are a way of showing this.
Yeah, thanks. Maybe just to comment that the whole field of cancer neuroscience is emerging now. It's subject to a grand challenge that's just been posed. So we welcome this in kidney cancer as well. So Scott, go ahead.
Thanks for the wonderful session, everyone. I had a question for Tom. So really intriguing data you shared about biomarkers of growth in small renal masses. And I wondered whether you've considered, again, like what is the underlying mechanism for increased growth? Is it a tumor cell intrinsic property that the mutations or copy numbers afford, or do you think it's some sort of environmental factor, blood supply, or some other stromal component, if you've looked at that?
Hi, Scott. Yeah, thank you very much. That's a very good question. We haven't looked in detail about the mechanism, and we haven't yet tied these findings with the microenvironment. That is going to be the next step. We're going to be doing that through both bulk RNA sequencing, but also 5K plex xenium. So hopefully we'll get an answer for that. And it'll be interesting to look at how the patient-specific risk factors will alter the selection of the genes that we see. So if you're a smoker, if you're obese, if you have a certain HLA typing, whether how much the microenvironment there then alters your gene selection. Thank you.
Maybe I can ask a quick question to Ari. So could you talk us through your sort of implementation plan for these signatures that you're now seeing as a more precise with regards to the TK partner?
Yeah, so as we start to develop better models to study because there's been so much work that have been done preclinically in TKIs on PDXs, which obviously have vascular supply but don't really tell you what else is going on. Maybe they'll have some limited myeloid populations, but as we start creating either GEMs or somatically engineered models, we're certainly working in the organoid space as well, we will have an opportunity to kind of look at the different TKIs, and so that's an ongoing effort that we have to compare different TKIs with each other to understand which particular populations. Many of the TAMs that are being affected are TAMs that are serving as antigen-presenting cells as well, so they are modulating T-cell responses. So you could imagine a scenario where you're depleting TAMs that are actually useful for immune response, and maybe that's why sometimes the long-term responses that we see don't exist in the TKIs, because over time, as the TAMs that are useful get depleted, they are creating a more adverse environment. So certainly that's where we're headed in terms of understanding that. And then yes, reducing it to specific biomarkers. You might maybe take a TAM population and then add one more marker onto it so that you could reliably determine what TKI, for example, a patient should get, or what drug you might want to target afterward. So.
I guess maybe one more quick question for myeloid. There's been this rich history of targeting myeloid cells, CSF1R. You've done some excellent work with the adenosine receptor. Where do you think the next generation for kidney cancer is going? Do you think it's specific TKIs? Do you think it's TREM2 depletion, something else in terms of myeloid targeting?
Yeah, no, that's really, so I think some of the, so it's a broad question, and yes, I think a lot of the myeloid strategies have failed because they're targeting a broad range of TAMs, for example, that are useful and pro and anti-tumor. So that, that's a sledgehammer, kind of like the Doge approach. Sorry to make a US reference. But kind of slashing everything when you should be, going very specifically. I think we're interested in sort of the cellular therapeutic angle. So I think really the goal would be to enhance the long-term memory responses, whether that's through depleting specific or enhancing TAMs that are promoting immune response, these antigen-presenting TAMs, or also other cellular therapeutic strategies that can enhance long-term memory. Because I think what we're seeing now, at least both peripherally and in intratumors in these patients that are home runs, are this long-term durable memory response that seems to be persisting.
No, I think we'll wrap up this session. Thank you so much to the panelists. This was wonderful.
Yes.